video on the chemical classes of hormones. So the purpose of this video is to sort of lay out what hormones are produced from, and then the chemicals that they're produced from, the molecules that they're produced from, and then how that relates to their hydrophilic versus lipophilic qualities. In other words, can they dissolve through membranes or can they not? Because that'll have play a big role in how individual hormones can affect cellular function, right? Because that's what hormones do, right? Hormones are chemical signals that alter the function of particular cells that express receptors for those hormones. Okay, so let's start. The first class of hormones I'm going to list here is amino acid derived. And what this means is that we're going to use specific amino acids as substrates, as the starting, blocks for starting substrate, if you will, for producing particular types of hormones. And there are really only, even though there are more than 20 or more amino acid structures, there are really only two that are utilized as a hormone precursor. And those are tyrosine and tryptophan. And really, we're going to focus more on tyrosine, right? And again, this means that there are the control of the hormone production is really control of the enzymes that act on these particular substrates, these two amino acids, to modify them to turn them into the hormones in question. So for the cells that are, making a particular hormone, they've got a specific group of enzymes to produce the hormones that cell type produces. And tyrosine can be used by two different glands to produce two different groups of hormones. And tyrosine is used to produce the catecholamines. And the catecholamines include, and they're actually made in sequence, one from the next, dopamine, which is then it can be converted into norepinephrine. Now, actually, dopamine is more commonly known as a neurotransmitter, but it is readily converted into norepinephrine. I'm going to abbreviate here, NE. And then norepinephrine is often largely converted to, and again, norepinephrine can also be a neurotransmitter. So again, the adrenal medulla, where these catecholamines are produced, would produce norepinephrine and epinephrine. Epinephrine, I usually abbreviate as epi. Right? It would produce those two, and it's going to that adrenal medulla will produce dopamine, most of which gets converted to norepinephrine, and then most of that gets converted into epinephrine. Okay, so it's just a sequential process, enzymatically tweaking the molecule to produce the hormones that are going to be secreted. And again, so specifically, or most importantly here, the norepinephrine and the epinephrine are known as our sympathetic hormones, right? Released in response to a stress response, or in a stress response. Okay, the other molecule, class of hormones, I should say, that tyrosine can be used to produce are the thyroid hormones. And those thyroid hormones are produced by the thyroid gland. Right? And they are, and again, just to give you the specific names, they are triiodothyronine, so that has 3 iodide molecules, and tetraiodothyronine, which has 4. Tetra means 4, right? And so those are our thyroid hormones, and actually the T4 is known as thyroxine, which is a more common term, even though that's not actually the active hormone. T3 is the active hormone. T4 gets converted into T3. Okay, but so the thyroid hormones. All right. Before we go any further, let's label these though as either hydrophilic or lipophilic. Put some colors on here. Right? And so we're going to label the hydrophilic hormones as with an H. Sorry, my email's popping up there. All right, I'm going to put a circle around this. Right? So all of the catecholamines are hydrophilic. The thyroid hormones are lipophilic. Okay, and again, we'll relate more about this later, but remember what that means is that lipophilic hormones, lipo meaning fat, philic meaning liking, are soluble in fats. So the thyroid hormones can dissolve into the cell membrane and passively enter or exit cells. Hydrophilic substances, including hormones, cannot pass through the lipid bilayer and would either need to be transported, which they typically are not, or they would need to bind a receptor that's actually at the surface of the cell. And again, we'll see that in a moment. All right, back to our other amino acid that's a substrate. And again, we'll focus much less on this one. And the reason for not really focusing much on it is that tryptophan is a precursor for melatonin. And melatonin has not been shown to have really dramatic physiological effects in humans, which is why we're not going to say too much about it. Light-dark cycle sort of entrainment, adapting physiological function to the light-dark cycle is a known function of melatonin in many species. You can actually buy melatonin at drugstores. They sell it as a sleep aid, right? Helping you to sort of to combat jet lag and things like that, because it's helping entrain what are called circadian rhythms or 24-hour rhythms. Okay, and I'll just throw this out because we were talking about it previously. Melatonin is actually got a separate indicator here. It's A for amphipathic. Amphipathic means it actually has portions of the molecule that are hydrophilic and portions of it that are lipophilic. And again, because we're not going to follow up on this much, we don't really have to relate that. But I did want to say that it's not lipophilic or hydrophilic. It's actually a little bit of both. Okay, so there is the first chemical class, the amino acid derived. The second chemical class that I'm going to lay out here is typically referred to as the peptide group. And that's a little bit misleading. So oftentimes it's simplified to call them the peptide group. I'm just going to list them all here. It's the peptide, protein, glycoprotein group. Right? And so these are the only hormones that are coded for by DNA, right? Because peptides, proteins, and glycoproteins are amino acid sequences. And just to remind you, I think we've heard this before, but glyco refers to carbohydrate. And so in that case, it's a protein that has a carbohydrate group attached to it. These are all hydrophilic. Okay, and again, we'll come back to why that's important in terms of the receptor binding. The only other thing to point out here really is that what's the difference here is that peptides are, by convention, defined as amino acid sequences of less than 50 amino acids. Proteins are more than 50, and then glycoproteins are more than 50 with a carbohydrate group. Okay, but not a lot of size and then adding carbohydrates are really the only difference here. And the last thing we'll add here is this group is actually most hormones. And this actually makes things a bit easier because what we're going to have you sort of require to know and use later on is, is a particular group of hormones hydrophilic or lipophilic, and then therefore how does it how can it affect cell function? And again, we'll get to that. But if we remember certain groups, right, then we remember all the others are going to fall into this, peptide protein glycoprotein, right? And we'll come back to that. I'll relate that when I get the last group out here. And that last group is the lipid derived. Right? And so there are actually two main lipids that are used to make chemical messengers. Cholesterol, and then another fat that's called arachidonic acid. Okay, and so cholesterol is actually both of these fats are found within membranes, and so that substrate is always present, and again can be just like group 1 here, the amino acid derived, can be enzymatically converted into these other molecules, these signaling molecules known as hormones, right? Well, and sometimes non-hormones, as we'll see in a second. But the cholesterol is the precursor for the steroid hormones. And those steroids come from a variety of locations. The gonads in male and female, right, for the estrogens and progesterone and testosterone, steroids like that. But then there are also adrenal steroids like cortisol and aldosterone. And so all of those are steroids. Actually, there's even a couple others. A calcium regulating steroid known as calcitriol. And one of the things you'll note about the steroids, and this is an important thing to remember, is that they tend to end in IOL or ONE, right? Corticosterone, aldosterone, calcitriol, right? They all sort of sound the same and end in that the same sort of sequence of letters, right? It's an easy way to sort of remember if it falls into this steroid class. And we're going to put an L on here because of course if you're fat derived, lipid derived, then you're going to be lipophilic. Now, going back to the last one here, the arachidonic acid is used as a substrate for a group that's called a really complex group that's called the icosanoids. And the good news is that most of these don't actually act as hormones. They can if they're secreted in really large amounts, but that's pretty rare. We will come across a few of them. They're a really important group of molecules. I'll mention a few things about them in a second. But they don't tend to be secreted in large enough amounts to act as hormones, meaning they don't get into the bloodstream in high enough quantities to sort of act at distant targets. Remember, we're using the classic definition of a hormone, which is a blood-borne chemical messenger. And the icosanoids include prostaglandins, and there's a whole variety of them. The thromboxane, and I'm sort of giving you the big groups here. This is a really complex group, but the big groups are the prostaglandins, the thromboxanes, and the leukotrienes. Okay, and the reason I want to point these out is not so much to study them as hormones, but to understand that some of the most common medicines that we take are used to block the production of icosanoids. Pain medications, many of them, the non-steroidal anti-inflammatory and pain meds like ibuprofen and Advil and things like that, block the production of the icosanoids. And the rationale there is that the prostaglandins are pain mediators and the leukotrienes are inflammation mediators. The other thing you may have heard of is people, especially old people, taking aspirin on the regular, and they do it because the thromboxanes are procoagulants, right? And so we want to block the sort of inhibit atypical blood clotting. And so people take aspirin every day to block production of the thromboxanes. So again, they're really important clinically, but again, not such a major player in terms of acting as a hormone. And again, because they're lipid derived, these are going to be lipophilic. All right, so how to remember these, right? So the key thing to remember is sort of like which are the lipophilic hormones, and then remember that everyone else is hydrophilic. So all you really need to remember, I'll put a little red star on them here, is that the steroids are lipophilic, and the thyroid hormones are lipophilic. And again, we said melatonin, not a major player, so we're not worried about that guy. We're not going to talk about the arachidonic acid metabolites as hormones. Should we know that they're involved in pain and inflammation? Sure, right? But we're not going to talk about them as hormones, so we can leave them out. So really, your lipophilic hormones are the steroids and the thyroid hormones, and everything else, the catecholamines and all of the protein, peptide, glycoprotein hormones are hydrophilic. Okay, now what does being hydrophilic or lipophilic mean in terms of how these molecules can affect cell function? All right, let's make some room here. And it's really going to be reasonably simple, because hydrophilic hormones are stuck in the interstitial space, right? I'll show you what I mean here. Right, so imagine we've got an endocrine cell here, and it's exocytosing its product. I'll put a vesicle releasing our substance. Like our substance, our hormone, red. Right? This is how many endocrine cells release their products, by exocytosis. Right? Of course, that's going to go into the bloodstream, right, if it's acting as a hormone. Drawing sort of this arbitrary set of blood vessels here, right? We would have diffusion into the blood vessel, and again, diffusion just about anywhere and everywhere, right, in terms of the interstitium. But that the hormone that gets into the bloodstream is going to be carried by the flow of that blood. It's going to end up in all tissues, or almost all tissues, where it has the potential to diffuse out. And so now we would have a target cell of some other type over here, right? And that cell must express receptors for this molecule, right? Now, if the molecule is hydrophilic, if we say that our hormone being released here is hydrophilic, right? That means water soluble. That means it cannot pass through the cell membrane here. Remember, when it leaves the enters or leaves the capillary, it's going between cells, right? That's a general rule. When it acts on a target cell, it has to do that by changing actions inside the cell. If it's hydrophilic, it cannot enter that cell. So our molecules are stuck out here in the interstitium. Well, if this cell has no receptors, nothing happens, right? If this cell expresses a receptor specific to this hydrophilic hormone, right, we're going to get binding, put one even farther into our receptor here, and then we can get action, right? It doesn't enter the cell, but it's going to be tied to other molecules, other proteins that are going to initiate action. Right? Hormones alter the types, amounts, or activities of cellular proteins. That's what they do. Proteins are the gears that make our cells do what they do. So altering the types, amounts, or activities will change the cell function and result in some sort of action. Okay, that's hydrophilic. If it's a lipophilic hormone, right? Let's put another cell over here. Right. There's a different endocrine cell. And actually, I'm not going to put exocytosing. I thought about it for a second. I'm not going to have an exocytosing because if it's a lipophilic molecule, it can actually just be produced and then diffuse out of this cell. So let's pick a color here for our hormone. Purple, right? And it would just diffuse out. And again, can diffuse any which direction, but we're going to have it, some of it diffuse into the bloodstream. Let's make it a little darker. Okay, and some of it's going to end up over here, right? And now I'll have it, it could affect both of these cell types, depending on if it has receptors. But let's add another cell type over here, right? Some crazy cell. And in this cell, right, now we don't have to have a receptor on the surface. We don't have to have a receptor on the surface because it's lipophilic, right? If it's lipophilic, right, this is the L1, this is the hydrophilic one, the letters on them here. If it's lipophilic, it can enter the cell. That doesn't mean it doesn't need a receptor, it just means the receptor can actually be found inside the cell. So in this case, now the receptor is in here, right? And I usually draw something. I had it up here as a V, okay, this is a little different shaped receptor, right? And our molecule, our hormone, can bind that receptor. Now, in addition, and let me erase and make this a little bit bigger. The receptor can be found within the nucleus. It could be here, right? And so our molecule can diffuse in, right? Here it's diffusing in, and it can bind a cytoplasmic receptor, or it could diffuse in and go through the nuclear and membrane as well and bind there, right? So the receptor could be either cytoplasmic or nuclear. The last possibility, and it's somewhat rare, it's a particular circumstance, but the last possibility is receptors found, this is supposed to be a mitochondrion, mito for mitochondrion, and we could have a receptor that's found here, right? Again, receptors for lipophilics can be inside of membrane-bound organelles because, again, our hormone has access to all of these things, right? Because it's lipophilic. You get to all of these locations. The only group that really does this are the thyroid hormones because they are involved in altering metabolism, and the mitochondrion plays a big role in the metabolic rate of our cells. Okay, so to summarize, our hydrophilic hormones, including the catecholamines and all the peptide protein, glycoproteins, will bind a membrane-bound receptor with a binding site facing outward because, again, the hormone is stuck out here in the interstitial fluid. Right, we're going to have to tie that to some actions inside the cell. Again, that's going to be in a separate video. Whereas if our molecule, our hormone, is lipophilic, as in other words, it's a steroid or a thyroid hormone, then it can bind a receptor in the cytoplasm, right here, the nucleus, right here, or in the mitochondrion down here. Right? And again, if it's in the mitochondrion, it's only the thyroid hormones, not typically the steroid hormones. And there we have it. We've got our chemical classes, and we've got how their chemical class determines whether they're hydrophilic or lipophilic. And then subsequently, now that we know that, we can tell you whether it binds an intracellular receptor or a membrane-bound receptor because we know whether it can enter the cell passively or if it's impermeable, the hydrophilic ones. All right.